Good afternoon. My name is Michaela Williamson, and the paper that I chose was titled HMGB1 Knockdown Increases Multiple Myeloma Cell Vulnerability by Regulating Autophagy and DNA Damage Repair. Here are the authors of the paper, and we will get started. So first, what is multiple myeloma? It initially begins as a benign condition called MGUS, which stands for monoclonal gammopathy of unknown significance. Where is multiple myeloma located? In plasma cells. So it inhibits the ability of the plasma cells to fight infections. And multiple myeloma is a hematological malignancy, which is very common, and it's second to lymphoma. There are less than 200,000 cases per year, and it is incurable due to drug resistance. These are what we know so far about multiple myeloma. This figure I have here just shows normal plasma cells and their antibodies, and the multiple myeloma plasma cells. So they're just abnormal compared to the originals, or the normal plasma cells. So there are three stages of multiple myeloma. Stage 1 is less aggressive. It might not be cancerous, it might be benign, could be malignant still. Stage 2, there's intermediate levels of cancer, so you could have a little bit, but not enough to form bone lesions. Or you could have stage 3, which is very aggressive. And at this point, it may start to cause issues with the bones and affect those, the kidneys, other organs. So the stages are very important, and they do determine the life expectancy or the survival time left, depending on what stage you have. So stage 1, the B2M, well, the beta-2M stands for serum beta-2 microglobulin. So that's going to be less than 3.5, and the serum albumin is going to be greater than or equal to 3.5. That is going to show a survival of about 62 months, could give or take. So stage 2, the beta-2M is going to be less than 3.5, but the albumin is going to be less than 3.5. Or the beta-2M is going to be 3.5 to 5.5, and the survival is going to be about 44 months for that. In stage 3, this is the most aggressive, the beta-2M is going to be over 5.5. And this survival is only about 29 months, so a little bit over 2 years, not very long. So the clinical features of multiple myeloma are known as CRAB. That's the abbreviation for it. So the C in CRAB stands for increased blood calcium levels, which is also known as hypercalcemia. Hypercalcemia can be caused by overactivity of the parathyroid glands or cancer. It just depends. The R in CRAB stands for renal insufficiency, meaning the kidneys lost some of their functions. They don't filter the waste like they should. They might not balance the fluids like they should or the right amounts. So the kidneys just aren't acting or functioning like they properly should. The A in CRAB stands for anemia, which could be two things. You could have reduced red blood cells, or you could have dysfunctional red blood cells. Either way, your organs are not getting the proper flow of oxygen that they need. And then the B in CRAB stands for bone lesions, which is what I have a picture of here. The bone lesions are just masses of tissue, and they could be malignant or benign. So in this picture, all these little holes, I've identified 3 right here. Those are going to be the bone lesions on this skull. So multiple myeloma and HMGB1. We know that multiple myeloma is drug resistant, and that HMGB1 causes the drug resistant cells. This is incurable, so even with chemotherapy or different drugs, they're not going to respond. The rapid development of including blood vessels are going to help the cells develop and metastasize, which is not good. Here are just multiple myeloma cells I have pictured. So what is HMGB1? I mentioned it previously in the last slide, but I didn't tell you what it was. HMGB1 is known as high mobility group box 1, or it could be known as high mobility protein 1. It is highly conserved, meaning it has decreased mutation. Once it's there, it does not mutate easily, and it's not easy to get rid of. It is a chromatin-associated protein that binds to DNA to stabilize the nucleosome. And with this, it affects arrangement, meaning it could unwind the DNA, fold it in different ways, fold it really tightly. It messes with the arrangement of the DNA. It also plays a role in DNA replication, DNA damage repair, and transcription. And here is that HMGB1 protein. So HMGB1 in cancer, it's involved in inflammation by upregulating pro-inflammatory cytokines and promoting carcinogenesis. Carcinogenic is just the normal cells transforming into cancer cells, which we don't want. Angiogenesis is the development of new blood vessels, and this also plays a role, along with invasion, progression, metastasis, and drug resistance. So in this figure, you can see the angiogenesis with a cancer cell. So here's the cancer cell, and here's the blood vessel supplying. And as more blood vessels develop and start engulfing the cell, you're providing more blood and more substance to that cell, allowing it to metastasize, allowing it to grow, which we don't want, especially since it is drug resistant. That's defeating the purpose. We don't want it to grow even further. Overexpression of HMGB1 is also something. It induces the activation of signaling pathways. And the HMGB1 binds to cancer cell receptors. And here I just mentioned what carcinogenesis is, like I just did, the normal cell to a cancer cell. So up here we have the HMGB1 using toll-like receptors to activate these signaling pathways. So depending on the pathway that is taken or what is activated determines the fate of that cell. So in previous studies, HMGB1 was looked at, and what they concluded was that it is a hematological malignancy and the HMGB1 is overexpressed, and some common ones are leukemia, lymphoma, and it is drug resistant, like I mentioned, and that is mediated through autophagy. And I thought this was super interesting. They're saying that HMGB1 kind of serves as a double-edged sword to the cell. So on one side, it protects the cells because it's drug resistant, so those chemotherapy drugs and other drugs aren't getting to the cell. But it also can cause the cell death once they go through the process, and that cell could die, just depending on the process that's taken and what happens to it. So in this picture, I just have the drug-resistant cell production. You have your autophagy inducers mixed with your pieces from other cells, and once that phagopore engulfs, it's going to engulf completely around the pieces of the cells that are mixed up. And once that's fully engulfed, it's going to form an autophagosome. And once that's mixed with the chemotherapeutics, that is where you get your multi-drug resistant cell. So autophagy, like I mentioned it briefly, is cell destruction or self-eating. The cell eats itself. It maintains homeostasis and protein degradation. And basically, it's a turnover of a destroyed cell organelles, and it's going to form a new cell. And depending on how much stress the cell goes under or what's happening to the cell if it's not used to it, that autophagy can be upregulated. So in this figure, just the autophagy process, you have the isolation membrane, and it's going to encompass or circle around or enclose in the cell debris that we've mentioned. So examples could be protein organelles, just anything like that. And then once it's going to fuse together, it's going to form that autophagosome like I mentioned. And then that lysosome is going to come in and bind to that autophagosome, forming an autolysosome, which is going to engulf that debris that's in that cell. Now looking at the mTOR pathway, it regulates autophagy. So when mTOR is activated, it inhibits autophagy. When mTOR is inactivated, it permits autophagy. So the autophagy pathway is going to regulate the fate of the cell, whether it dies or whether it survives. So in this figure, you can see mTOR here in the middle. Once a signal starts this pathway, depending what is activated or not, will determine if mTOR is activated or not. So if mTOR is activated, autophagy and caspase-3, which leads to apoptosis, are going to be inhibited. So they're not going to be allowed. That cell is going to remain how it is with mTOR activated. When mTOR is not activated, autophagy and apoptosis can be permitted as normal, and that cell could die. So HMGB1 with apoptosis. Apoptosis is cell death. So apoptotic cells, they atrophy macrophages. And a macrophage is a phagocytic cell that just ingests foreign materials that it's not used to. And it consumes drug-resistant cells. So toll-like receptors release HMGB1, which then makes that cell no longer drug-resistant since that HMGB1 is released from the cell. And now that cell can die due to chemotherapeutics since the cell is no longer drug-resistant. So in this figure, it's just the apoptosis cycle. Starting with a normal cell, it shrinks, the organelles disintegrate, the membrane continues to bleb, and the organelles and nucleus collapse, and the apoptotic bodies form. The macrophage comes in since it's foreign material and engulfs that, and that is your apoptotic cycle. So now looking at DNA damage repair. There are different kind of repair mechanisms depending on the type of damage that's seen. So the damage is identified and then corrected. So with single-stranded damage, there are different types of ways you can handle this. You could do reversal, mismatch repair, base excision repair, and nucleotide excision repair. With double-stranded damage, you have two options: homologous recombination or non-homologous end joining. So the HMGB1 binds to DNA like I mentioned previously. So when it binds to that damaged DNA, it's going to further damage it, and it's going to further bend it, leading to more issues. And for this figure, I just picked the single-stranded damage pathway of nucleotide excision repair, showing the damaged nucleotides being hit with different type of ultraviolet rays, which then takes out those damaged nucleotides along with a couple others that weren't damaged, but just since they were so close. That polymerase comes in and adds the correct nucleotide sequences that are matched with the other nucleotide sequence, and that ligates them together, creating your new DNA sequence. So the overall paper, the main question is, what is the role of HMGB1 in multiple myeloma cell proliferation and drug resistance? First, we're going to discuss the cells that they used. So they used human multiple myeloma cell lines, and they used primary multiple myeloma cells. The human cell lines used were RPM 18226, which is a myeloma cell line, CAG, which is a plasma cell myeloma line, and MM1S, which is just a cancer cell line. Bone marrow tissues were also looked at. This was because with the bone marrow, if the multiple myeloma cells are outside of the bone marrow, then surgery could be used. If the issue is within the bone marrow, that is when there's more issues with that, and surgery cannot be done. So that's what they wanted to focus on in this paper and in this research. They also used CD138 positive cells. So since these are CD138 positive, they do acquire the expression of CD138, which is also known as Syndikin-1 or SDC1. That is a marker for terminally differentiated normal plasma cells. So these cells do show the differentiated normal plasma cells. And this picture just shows where the bone marrow is located within a bone, and that's where you would find the plasma cells that are normal or the multiple myeloma cells that are causing issues. So RNA interference was used with short hairpin RNA against HMGB1. What they did was they transfected a lentivirus that contained short hairpin RNA that's against HMGB1 into multiple myeloma cells. They also used a control lentivirus. A lentivirus is just used to cause chronic and deadly diseases, so they wanted to put that in that cell with a control and one with HMGB1 to look at the effects of that. And when they used the RNA interference, the result of that was degraded HMGB1 cells, since that was what was in the lentivirus, And then the messenger RNA levels were detected using gene array, not RNA, sorry. And here I just chose the RNA, a super simple explanation or pathway for the RNA interference, just showing that the double-stranded RNA is cleaved by the dicer complex and forms siRNAs. RISC comes in, that single-stranded siRNA interacts with RISC, which then creates some foreign RNA. Since it is foreign, it is degraded, and that RISC is going to come back and act again. QRT-PCR was also used, which stands for Quantitative Real-Time Polymerase Chain Reaction. What this is you amplify DNA products, and when they amplified those products, they measured them after each cycle. In order to amplify it, you need certain things to be added. So you need the PCR reagents, you need specific primers, and the fluorescent molecule. The main advantage to the quantitative versus qualitative, the quantitative shows the initial amount or number of template DNA copies, where qualitative does not. So that's why they chose to use the quantitative. So here in my figure, you see that you start with a sample, you extract the DNA, you add the PCR reagent, the primers that they wanted to use, and then that fluorescent molecule so that they could see what they were getting. They amplified it, and then they detected it, and then they analyzed what they detected to get their results. Western blot was also used of multiple myeloma cells. And at this point in the semester, I think we all know what Western blot is, but I put a figure up and a slide just explaining it. So you're gonna, what Western blot does is it shows the extraction of a total soluble cellular protein. So you start with gel electrophoresis. Then the proteins are separated using SCS page, then transferred to PVDF membranes, and you want to block specific proteins. You only want to show the one that you're looking at. So to do this, you're going to use a primary antibody and incubate it, and that targets the protein. And then you're going to incubate it with a secondary antibody, which targets the antigen. Then you're able to detect and analyze. Cell proliferation assays were also used. So multiple myeloma cells were plated in 24 cell plates, or well plates, with or without dexamethasone. Dexamethasone, or also known as DEX, which is what I will be referring to it as, is a glucocorticosteroid that treats inflammation. So once they were either with or without DEX, they were transferred to 96 well plates, and the absorbance was measured at 490 nanometers to check the cell viability. And here just shows those adding agents and then measuring the fluorescence of them. So then they also wanted to look at the apoptosis, cell cycle, and autophagy detection of the cells. And to do this, they used flow cytometry. So the apoptotic cells were either incubated with DEX or not. So the ones incubated with DEX were either with or without CQ, which means quantitative cycles. The level of apoptosis was measured using flow cytometry. And like I said, same with cell cycle and autophagy. So what they did was the samples were suspended in the sheath fluid. And then single file, they go through the flow cytometer. This laser shoots across, and that's how they detect the cells, where are they at in the cell cycle and autophagy and all that. So this was also used. They also used the NOD-SCID mice, which were subcutaneously injected. After 1 week, the mice were divided into 4 separate groups, and some were injected with DEX, some were not. Tumor measurements were then taken using a certain formula that they came up with that's in the paper, and then the volume was calculated of those tumors. The samples of the tumors were extracted and put in a 4% paraformaldehyde, and then immunohistochemical staining was done. And here is just a NOD-SCID mice with a tumor on the right flank, which is where these mice in the experiment were also injected. So the first question that they asked was, do multiple myeloma cell lines and primary multiple myeloma samples express high levels of HMGB1? So the expression of HMGB1 in multiple myeloma cell lines, MM1S, 8226, ARK, ARP1, H929, and in primary multiple myeloma samples are shown here. So they all express Here's the different lines that I just mentioned. And this is just the gene expression. So each one of these cell lines do express it. The messenger RNA levels were then measured using qRT-PCR. And then the protein levels were analyzed with a Western blot as we see here. And this does show up here. This is the HMGB1. Down here is the cell lines. So HMGB1 was expressed in both. So then immunofluorescent analysis of bone marrow tissue was done using the CD138 positive plasma cells, which expressed HMGB1. And then they used Oncomine data, which is just a database to further look at more information. Once they received their results, they wanted more, just to make sure they were on the right page. And that did show the expression of HMGB1, but it also shows that it was negatively associated with the survival of the multiple myeloma patient. So A and B up here, the same pictures before, I just wanted to re-put them on here to show that it was expressed still. HMGB1 is still expressed in figure C, showing the same thing as pretty much figure A does. Just with a couple more of the cell types. But then figure E shows this is when they're alive at 3 years, the survival of the multiple myeloma patients, and then dead at 3 years. They're about the same, showing there's a negative association with the survival and the expression of HMGB1, even though it's expressed, that has nothing to do with this patient's survival. So then down here, this is where you're going to look at the immunofluorescence of the bone marrow and the different cell types with HMGB1, and then when it's merged together, showing all that in one. The second question, does HMGB1 knockdown in multiple myeloma cells enhance the inhibitory effects of chemotherapy with DEX via apoptosis? The multiple myeloma cells were transfected with HMGB1 knockdown lentivirus, like I mentioned, and this shows that HMGB1 was downregulated. So when you look at the cell line 8226, CAG, and MM1S, the first row up here is the control, the second is the one containing HMGB1. The control shows more than the HMGB1 in both these pictures, showing that HMGB1 was downregulated. But this third picture threw me off a little bit because to me it looks like the HMGB1 is upregulated, so I don't know if they didn't catch it, if there's a picture issue, I'm not really quite sure, but based on their results, the HMGB1 was downregulated. So then looking at figures B and C, the first box is the control, the black is the HMGB1 knockdown, so that was decreased or downregulated. And then when using Western blot, you have your control versus your HMGB1 knockdown, and in each of these cell lines, once again it shows that it is deregulated or downregulated, showing the same results across all of those. Proliferation between the knockdown and control multiple myeloma cells shows that it significantly increased the inhibitory effects of DEX and the HMGB1 knockdown compared to control. So when looking at figure D, it shows that the bottom line here, and this one you can kind of see here, and then it's a small bit over here, that those lines with the HMGB1 is lower. So the cell number decreased. So there is an increased inhibitory effect with the DEX. And then the same with E, the bottom shows the HMGB1, in both figures, and the top is the control, showing that there is increased inhibitory effects. The third question is, does the downregulation of HMGB1 activate the mTOR pathway, inhibit autophagy, and increase DNA damage when induced by DEX? When they looked at this, they found that there was no differences among the cell cycles in the two groups. So when you look at these pictures, the gray in the bottom of these is the G1 phase of the cell cycle, the black is the S phase, and then the white is the G2M phase. So among these, this is a control, this is a knockdown. They show the same. Control, knockdown, about the same, and so on. So there is really no difference in the cell cycle. And these are two different cell lines here. And then this is showing that this is a control without DEX, control with DEX, knockdown without, knockdown with, basically just showing a little bit more expression, but about the same in regards to the cell cycle. The HMGB1 expression was suppressed and increased DEX-induced apoptosis of the multiple myeloma cells. So when looking at figure C, the H226, CAG, and MM1S cell lines show that with DEX, the cell apoptosis in the knockdown was increased compared to the control. And that just shows that the apoptosis wasn't increased when DEX was induced. To further look at this, they wanted to use Western blot. And this shows that there's increased levels of DEX-induced cleaved CPARP, which CPARP stands for C-capase 3 and poly-ADP ribose polymerase. Up top shows your PARP and CPARP, so the CPARP is the second row. So it's showing as you move across with the DEX being induced and the knockdown, it's increasing. So it does increase the cleaved CPARP as you move across there. And then same with this figure, it does show that it does increase when the HMGB1 knockout or knockdown is induced with DEX. The fourth question is, after treatment with DEX in xenograft models, does the tumor burden of the HMGB1 knockdown mice decrease compared to the control mice? So by looking at this, they wanted to see the decreased levels of survival of When looking at this, it showed decreased levels of survival and apoptosis related genes. So the survival would be DEPTOR, which is a domain containing mTOR, and then the apoptotic genes were BCL2, BCLXL, and MCL1. They observed these through an Affin-Matrix HTA 2.0 array, which just compared the different changes. So down here you see the 8226 cell line and CAG, and this is with the HMGB1, and this is control. This is knockout HMGB1 and control. Just shows the different levels of these genes and where they are present and where they are not. And then they wanted to further verify those results using the QRT-PCR and Western blot. So when they did this, they found that the anti-apoptotic and multiple myeloma cell-related, the survival-related genes decreased in the knockdown when compared to the control. So in figure B, you have the DEPTOR gene, BCL2, BCLXL. And each one of these genes, it shows that in the knockdown, which is the black, it is decreased compared to the control. So that gene expression of the HMGB1 is decreased. Then they wanted to look at a Western blot, and it does show in each one of these different genes and each one of these cell lines that it is decreased when the knockdown is present. And same with figure D. It's showing that across the board, when HMGB1 short hairpin RNA is present, that there is a decrease in the knockdown compared to the control. So then looking at the mTOR pathway activation, when looking at DEPTOR gene, it's downregulated after the HMGB1 knockdown. And then when looking at the phosphorylation of the P706K and AKT SIR473, there's an increase in HMGB1 knockdown multiple myeloma cells, and the control cells are really unremarkable, so it's hard to just see them. So the pathway is activated after HMGB1 knockdown. And across all the board, you can see that the first one is a control with no DEX. Next is knockdown without DEX, control with, knockdown without. Just showing that the downregulation after the HMGB1 knockdown, and then the increase after that knockdown when you're looking at the Western blotting dealing with the phosphorylation. So then when you're looking at the DEX exposure, so when they were exposed to DEX, there was a decrease of LC3AB11, and then that was autophagy induced. So when you look at this figure, you can see the first is control, second is knockout. They're induced with DEX. And they decreased. So there's a decrease of the LC3AB11. But then when you look at the apoptosis, it increased in the RPN188226 cells, showing that there's an autophagy inhibitor present. So in each one, you start out with the control, and then you have the knockout, and that increased in those cell lines. So HMGB1 does induce multiple myeloma cell drug resistance via autophagy. When you're looking at DNA damage, the RAD51 expression was observed, and that's involved in the DNA damage repair pathway. And that significantly decreased. So as you look at the RAD51 expression, it decreased as DEX was added and the knockdown was used. And same with the MM1F cell line. Then when looked at the gamma H2AX, it's a DNA damage marker, that increased. So when you look at the control versus the knockdown, the control increased because the HMGB1 was silenced. And same with the MM1F cell line again. So the RPN18226 cells, when apoptosis occurs, the DEX is present, and then when the AZD6738 is present, that's the DNA damage repair inhibitor. And then this Western blot concludes the same with the gamma H2AX and the HMGB1 being increased, and then the expression of RAD51 being decreased. So then when looking at the tumors from those non-skid mice, the sensitivity enhanced in the multiple myeloma cells were looked at in DECTOR and BCL2 genes, and those were decreased. So then when the non-skid mice were looked at again with the RPM18226 cells that were present, and those treated with DEX, those mice showed a lower tumor burden than the other mice did. So this first row up top is the control that did not have DEX. The second is the knockdown containing HMGB1 without DEX. The third row is the control short hairpin RNA with DEX. And the last is the knockdown containing HMGB1 with DEX. As you can see, when DEX is added, these tumors are a lot smaller. So the tumor burden was lower, and when DEX wasn't induced, they were bigger. So that sensitivity was decreased. So from all of these results and everything I've learned and read in this paper, it was concluded that HMGB1 does play a role in autophagy and DNA damage repair. The downregulation of HMGB1 enhances cell sensitivity to DEX, and HMGB1 may serve as a target for multiple myeloma treatment. There just needs to be further experiments done and more research done behind this to really further know. There were a couple future research questions. The first one is, what's the mechanism by which the HMGB1 regulates protein expression? We know that it does regulate it, but what's that mechanism that actually does this? The second is, what's the effect of HMGB1 on DEX-induced mTOR pathway activation? We know it has an effect, but what is that? Like, what is the exact effect on that pathway activation? And the third is, HMGB1 knockdown may inhibit repair of replication-dependent DNA damage. They believe that it may inhibit, but they're really not sure, so more research needs to be done on that to actually see if it does. And here is my reference. Thank you.